All right, so thanks for the introduction and thanks for the invite out. It's been a fun morning so far, and I look forward to meeting everybody in the afternoon. I even actually discovered that I went to elementary school with one of your faculty, halfway or all the way across the country, actually. So that was kind of a cool discovery. All right, so see if I can get this working. Sorry, give me one sec here. Okay. All right, good. Otherwise, I would have been pinned behind the podium the whole time. Thanks. Okay, so as Dobie was mentioning, so we study quite a few different kind of directions in the lab. So we do a lot of work on selective attention, perceptual decision making, kind of the interface between those two things. And then more recently, we've been studying working memory and also how those relate to things like selective attention or selective information processing. And we use a bunch of different tools. So we do a lot of EEG work, a lot of fMRI work, and a lot of basic psychophysics, and then try and kind of link those with some simple models to kind of bridge the levels of analysis. So today, I'm going to focus on kind of one subset of this work that focuses on working memory using fMRI as our kind of major tool here. And before I get going, I'd actually like to emphasize that clarification questions are very welcome. So there's a couple of spots during the talk where I might drop people. And if I do that, please stop me and just ask me to try and explain it a different way, or Dowie can explain it to you for me. And hopefully that way we can try and not drop too many people here as we're going through. But that's definitely welcome, so please just speak up. Okay, so when we talk about working memory, we're just talking about remembering visual information in our case. It could be auditory, but we focus on the visual domain. But remembering information for a very short period of time, so typically on the order of a couple of seconds. And you want to be thinking about this as kind of a model system for how you find your car keys on the couch when you're in a hurry in the morning and trying to get out the door. Okay? And so that is, how do you hold information in mind, like an image of something, and use that to guide behavior? And we do this using pretty simple tasks. And so this is just kind of one canonical paradigm that's used in the field. And in this type of experiment, what we do is just ask people to remember a bunch of colored squares, or it might be line orientations or something like that. We take that away. There's a short blank period, let's say a second or two. And then we put up a test array after that. And we just ask the subjects if those two displays are the same or if they're different. Okay? And so in this case, the answer would be different because this purple square here changed to a red square there. Okay? But the subject's task, again, is pretty straightforward. They're just saying same, different on these two arrays here. Now, what you see is that behavior varies very systematically in these tasks. So if I ask you to remember one colored square, and then I ask you if it's the same or different, that's trivially easy, right? If I ask you two colored squares, also easy. Three colored squares, pretty easy. But then when you start getting above three colored squares, performance in this task tanks pretty quickly, okay? And it turns out that this is actually a very stable trait for a given individual. So I could run this simple memory task on you, take you back a year later, and your performance would be largely unchanged. Okay? And furthermore, what we see is that there's a lot of variability between people. So some people really struggle to remember, let's say, half an item down here. I'm not sure what's going on with those folks. But other people can remember, 4 items easily, and if you were to expand this out even farther, you see that some people go up to 5, 6 items. Okay, so some people can remember a lot of information with a lot of ease. And one of the reasons why this is so interesting and important, and we think it's a good topic of study, is that performance on those really simple tasks like I just gave you, like how many colored squares can you remember, predicts things like general intelligence, success at high school, GRE scores, pretty much any outcome measure you can think of in that domain. Okay? Even more interestingly, from my point of view, is that it also predicts things like the efficiency of attentional control. So how well can you select relevant information and ignore irrelevant information? That's very strongly linked to how much colored squares, silly as that sounds, you can hold in memory at a given time. And so we think that using these very simple tasks allows us to tap into something that's really a core cognitive function and that relates to a lot of your other capabilities in terms of kind of higher order cognition. Okay, so today I want to walk through a set of studies that's actually taken place over the last 10 years or so. And I'm going to start by setting up kind of two hypotheses at either end of a spectrum here of hypotheses about how you go about remembering visual information for short periods of time. Okay? And on the first, or kind of on one end of the spectrum here, I'm going to talk about what's called the sensory recruitment hypothesis. And this is just the idea that you use, the way you remember things is just by using the same neural machinery that you used to see them in the first place. Okay, so in the visual domain, let's say you process information in early visual cortex, you would just kind of reinstantiate those same patterns of activity that are present when you're seeing things and hold that in a kind of tonic fashion during a delay period or during the time you're trying to remember stuff, and then rely on that to support your ability to find the thing that you're looking for in the environment. Okay? And so that's one kind of idea here. And one of the interesting kind of corollaries of that idea is that the way you remember things is by using or co-opting these sensory mechanisms. So that means the neural code for a memory is the same or is very similar to the neural code associated with seeing it in the first place. Okay? Now, another idea that's especially gained prominent, I mean, like all ideas in neuroscience and psychology, like waxes and wanes over time. But this idea has come back into fashion recently. And this is basically the opposite of the sensory recruitment hypothesis. And it says, okay, so you're using your eyes to see, right? And you get some information you want to remember, but then you're looking around, let's say, for your car keys. And every time you make a saccade, every time you move your eyes, you get a new batch of visual information coming in. It's going to flood primary visual cortex with activity. And if you're trying to remember stuff using that same chunk of tissue, it's going to get overwritten. And so this idea of sensory recruitment is just kind of silly and shouldn't really be the way that the brain's engineered, right? And instead, what you should be doing is, seeing things, taking that information in and recoding it into a non-sensory-like format and storing that information via persistent activity elsewhere in the brain. Okay? And where that elsewhere is, that depends on who you ask, parietal cortex, prefrontal cortex, and so on. But for my purpose, the key kind of notion of this model is that you're recoding the information out of a sensory-like format and into a format that's not likely to be interfered with by new information that you're processing. Okay? And so I just want to really emphasize that point that distinguishing between these two ideas is more than finding out whether memories live in V1 or they live in wherever your other favorite brain area is. It's really more about how the code or what the format of the code is that supports your ability to remember information. Yep.
Just really quickly, you showed an example that was a single stimulus. Does anything change about this if you're talking about sequences, so the temporal dimension? What you're talking about?
So a temporal, yeah, so that's a good question. Maybe I'll defer on that for the moment, but there is some evidence that you're predictively reinstantiating things in early visual cortex to support memory. Yep. Okay, cool. Any other questions before we kind of launch into things here at the moment? Okay. Oop. All right, cool. Okay, so what I want to do is start by providing some evidence for this hypothesis, and then I'll kind of provide some evidence for this hypothesis, and then we'll kind of wrap back around and see where we end up at the very end here. All right, so anyway, about 10 years ago or so, we got started down this pathway of trying to figure out whether the sensory recruitment hypothesis was viable. And fortunately, it makes a very simple prediction, right? So if you're using the same cortical areas to see and to remember stuff, then we should be able to look in early areas of visual cortex, and we should be able to tell what you're remembering based on activity patterns in early visual cortex, even after we take a stimulus away. Okay? And so just as kind of a simple example here, let's say I ask you to remember the orientation of a line or a grading or something like that. We know that V1 is specialized, presumably, for processing that kind of information or for decomposing that information out of the environment. So we would expect to see early areas like primary visual cortex that have a lot of orientation selective cells maintain that activity across delay periods during a memory interval. Okay? So to test this idea, we designed a variant of the task that I showed you at the very beginning. It's kind of more complex in some ways and simpler in other ways. But let me walk through it, and then we can ask questions if people have any at the end here. So at the beginning of each trial, subjects see a display that looks like this. And so it's got a grading that's either an angle right around 45 degrees, like this, or an angle right around 135 degrees, like that. And it's some shade of red or some shade of green. Okay? And then we ask the subjects on a given trial to either remember the exact shade of red, let's say, or the exact orientation here. And by that I mean, is it 46.5 degrees or is it 46 degrees, that kind of thing. We take that away. There's a 10-second delay period during which time there's no external stimulus coming in, so there's no more sensory drive. And then we put up a test stimulus at the end, and we either say, is this the exact same color, so the exact same shade of red, for example, or is it the exact same angle that I showed you at the beginning? Okay? And so they're basically just doing a 2 alternative force choice task. Only orientation or color is relevant on a given trial. And we just kind of cue them which feature to remember on a trial-by-trial basis. Okay? And doing this, we can also adjust the difficulty of the task so that it's matched across all the conditions. So we can change how subtle we can manipulate how subtle the changes between the feature that they're remembering and so forth to control behavior in that way as well. Okay? Yep.
Are they cued on the relevant feature during the sample phase or during the test phase?
So 10 years ago, but it's either right before the start of a trial or right at the beginning of a block. But they definitely know going into the trial what they're going to have to remember. Yeah, and I'd have to look back to see if we did it block by block or trial by trial. But, yep. But yeah, that's a really key point. Thanks. So any other questions about the paradigm? Yeah, go for it. Yeah. So before they see the sample stimulus, they know if they're going to have to remember the color or the orientation. And I'll say a few more words about why we did that in a few minutes when we get to the data, but yeah. Okay, cool. All right, and so, given the simple task, then what we're going to do is say, okay, so if we read out the information using fMRI and primary visual cortex, can we tell whether they're remembering, an angle around 45 degrees or an angle around 135, or whether they're remembering a red or a green? Okay? And so that's kind of the basic, where we're going with the analysis at least. Okay, so the analysis itself here, maybe some of you have gone through this, but if not, I'll kind of walk through the logic. So in primary visual cortex, right, so we've got columnar organization, you've got orientation columns and some color selective stuff kind of intermixed in there. But these are very, very small. So the columns are sub-millimeter. With an fMRI voxel, so a single volumetric pixel here, we're sampling somewhere on the order of 8 cubic millimeters, oftentimes more like 27 cubic millimeters, but much, much bigger spatial, or much, much coarser spatial resolution than the underlying kind of functional organization that we want to tap into, right? And so what happens, though, is that if you look and you make enough measurements and you really kind of dig in, these fMRI voxels will often exhibit subtle feature preferences, presumably due to the composition or the distribution, let's say, of underlying feature selectivity at the neuronal level or at the columnar level. And so, for example, if all of these columns go into that voxel may exhibit a slight orientation preference for something angled about here. Okay, and at the single voxel level, these effects are not, these selectivity is not huge, but it's reliable, and I'll kind of explain how we can extract that information in a minute here. But that's the basic logic. And then the next step, given the weak selectivity of these voxels, is just to exploit the fact that we have 1,000 of them in primary visual cortex. Okay? And so what that means is that we can show a stimulus like this, let's say like a horizontal grading, and we can learn the pattern of activation across all of the 1,000 voxels in V1, and we can say okay, that voxel responds a lot to horizontal. We can show another angle like this. We get another pattern of activation out, and we just train support vector machines or any other kind of simple machine learning algorithm to recognize these patterns associated with changes in the orientation of the stimulus. Okay? All right, so questions about that, or good so far? Okay, cool. Okay, and then once we've learned those patterns, of course, we can then guess based on a novel pattern of activity what the subject was remembering. Right? And so here's the data from this first study, or half the data here. So this is how well we can classify the orientation of the stimulus. So is it 45 or is it 135 degrees? When they're told to remember the orientation or when they're told to remember the color. Okay? And what we see is that we can significantly classify the orientation they're remembering when that feature is relevant, but not when the color is relevant. Okay? And we see the opposite pattern when we try and classify color. We can only classify it when they're told to remember the color and not when they're told to remember the orientation. Okay? And so what this means is that our ability to classify that information is not just due to a passive sensory response, right? Because on every trial you've got a color and an orientation. But it's really yoked to the feature that they're trying to remember during that 10-second delay period, because that's the only thing we can pull information out about in visual cortex. Okay, okay, so questions about that so far? Yep, go for it.
So I get the orientation classification from the fMRI data, but how did the color classification work?
Same way, so same general logic. We can train classifiers to recognize patterns of activation associated with different colors. And V1 has, actually, I mean, even in the classic textbooks, it's got a lot of color selectivity, but it's probably even more prominent than we've realized in the past. So, yeah.
So the stimulus that you give, do they have both the features when you're training, or will they have just one?
So in this case, the model was trained based on the like a hold one out procedure using the memory data itself. And then we all, actually, the next thing I'll show you here is that we all, actually let me just show you and then you can go on, so cool. Right, so that brings up a good point. I actually kind of glossed over this, but these models were all trained by learning the patterns of activation during that memory delay period on some training data and then applying it to held out test data during the memory task. But again, the sensory recruitment idea is that we should see the same patterns of activity during viewing a stimulus as we do during a memory delay period. And so the next thing we did was we trained another classifier based on the presentation of one of these colored gratings, and it could be red or green or 45, 135, et cetera, that we just blasted at the subject for 10-second blocks. And so we just present this over and over again, then change it, and so forth. But anyway, so we trained a classifier based on this pure sensory task, no memory demands at all. And then we generalized it to the memory delay period, and that also works. So I'm just showing you orientation here. It works for color as well. But again, this demonstrates that there's at least enough similarity between the patterns of activation evoked during a pure sensory task and during this memory task that we can pull out the feature that the subject's remembering. Okay. So just to kind of quickly summarize this one. So, what we've done is taken kind of the first step towards supporting the sensory recruitment idea, right? And so, we've shown that there are sustained activation patterns in early visual areas that resemble the patterns that you see during sensation, or during actually seeing the stimulus itself. And so this suggests that it's at least plausible, right, that this kind of a mechanism might be helping to support your ability to remember things for short periods of time. Okay, so anyway, so that was the first step. So the next thing we wanted to do is see if this is actually functionally significant, right? And so fMRI, of course, is not a causal tool at all. But the next kind of thing that we can do logically is try and link the quality of the activation patterns, or the amount of information, let's say, in early visual cortex with behavior during these memory tasks. And so to do that, we designed, again, in even simpler tasks, there's just kind of one twist on it. In this case, subjects just have to remember an orientation, so just a single grating, and they are remembering its tilt. There's a delay period, again, no sensory stimulation here, and then we give them a recall task at the very end here. So by that, I just mean we put up another grating that's randomly oriented with respect to the first grating here, and we have them rotate it until it matches the remembered orientation of that stimulus. Okay? And the reason we do that is it gives us a much finer measure of the behavioral precision of this, or the mnemonic precision of the subject, right? And so if they perfectly match this grading, that means they remembered it, with perfect precision. But to the extent that they're off, that gives us an idea about how accurate they are across all the trials. Okay? All right. Okay, and then the next thing we did since we're now dealing with a kind of a continuous stimulus space here, so any orientation, we could ask them to remember any orientation. We also adapted the way that we're analyzing the activation patterns in early visual cortex to produce a continuous output, or kind of a continuous estimate of what the stimulus is that they're holding in mind. And so the way we do this is by modeling the response of each voxel in V1 using a set of 9, or actually the exact number doesn't really matter a whole lot, but let's say 9 different orientation-tuned filters here. And these are just kind of meant to superficially mimic what you might see at the underlying population level. So we've got these kind of circular Gaussian functions that are arrayed across different points of the relevant space that we're trying to model here, which is orientation. Okay? And so basically what we're going to do in the first step here is just estimate the selectivity or the responsivity of each voxel to each of these different orientations using a simple linear model kind of framework. Okay? All right. And so we go through and we do that for every voxel in V1. And so, you know, voxel 1 might have some weight profile, voxel 2 will have a different one, and so on. And we just do that for all 1,000 voxels that we've got in primary visual cortex. And we do this also, of course, using just part of the training, or part of the data rather. So we've got a training set that, you know, we hold out and we just use to train the model here. Okay? All right. Now, in the second part of this analysis, what we do is we take a pattern of activation across all the voxels in V1. So that's what we're looking at here. We've learned how selective each voxel is for each orientation, right, in the first part. And that allows us to then say, okay, we've got an observed activation pattern. We know the selectivity of each voxel. What must the stimulus have been that evoked this pattern of activity? So we're basically like inverting the linear model and going back into stimulus space. And this is actually, I mean, you can think about this as a reconstruction of the stimulus, of our estimate of what the stimulus was, right? Okay. Now, we do that on every single trial. And of course, you know, sometimes they're remembering that angle, sometimes this angle, and so forth. So we then take all of these reconstructions and we recenter them based on the remembered orientation here. So what it should be peaking at. We're not taking a max rule here. We're centering it on the actual orientation they should be remembering. And then by convention, the data I'll show you are all centered on 0 here, where 0 is the orientation they were supposed to be remembering. Okay? Okay, so questions about that? This is actually one of those spots where if I drop people here, the rest of it's going to unravel really quick. So does that kind of make sense? So, I mean, you can think about this as just kind of another representation of the stimulus that they're remembering, and it's our estimate of that stimulus. Okay, okay, and then I guess the last thing I would say that's kind of important to keep in mind is that if V1 had perfect information about the remembered feature, this would approach like a stick function, right? And if there's no information at all, this is just going to be a flat line. There's going to be no relationship between what we're reconstructing and what the actual remembered stimulus was. Okay? All right, cool. Okay, so just to remind you here, we've also got a recall task, right, in this case. So we also have a continuous kind of graded measure of how precise each subject was. And so here's the data from a subject here. And basically what you can do then is say, okay, if the subject was perfect on every single trial, then their responses, their error in this behavioral recall task would be clustered right at 0, right? And to the extent that they are not remembering, and let's say they were doing it with their eyes closed, this would be a uniform distribution, right? They would just be randomly guessing, okay? Okay, and so now what we've got is a nice kind of graded measure of behavior and our stimulus, or our reconstruction of what we think they're remembering, and then we can try and link those two things together, okay? All right, and so the way we do that is we just take our orientation reconstruction. So this is, you know, our estimate of what subjects are remembering based on activity, I guess, actually in V1, V2, I suppose. And then we can estimate the precision of this, or kind of the fidelity of it, just by, actually we can do it a bunch of different ways. I think in this study we probably fit a circular Gaussian to it and measure the bandwidth of it. But basically we just want to know like how flat this is or how peaked it is, right? As our measure of the precision, or our measure of how much information is in V1. And then we can also, I'll just jump back here. We can also do the same thing for this. We can just measure, you know, the standard deviation of this distribution as our measure of behavioral precision. Okay? And if we link these two things together, what we see is that there's a strong correlation across subjects here. So we've got mean absolute recall error here on the bottom, which is big numbers are bad here. That means, you know, you're more dispersion about 0 in your responses. And then this is the dispersion of those reconstructions, where again, bigger numbers here means flatter functions or less information in primary visual cortex. Okay? And this also works within subjects. So on a trial-by-trial basis, we can go through and we can say, like, okay, what's the precision of the reconstruction of the remembered thing? How well do you actually do in the behavioral task? And we've also linked this in other domains like spatial working memory and a few other times where measures of behavioral precision have been linked to the amount of information we can extract from early visual cortex. Okay, so in this second study, right, so kind of replicates the first in that we're finding information in early visual cortex about remembered features, which again is consistent with the sensory recruitment hypothesis. And then in the second study here, we also took kind of one additional step in terms of relating in a correlational way the activation patterns or the quality of the activation patterns and the behavior of the subject. Okay, so again, we don't really have the tools to properly do this in a kind of a causal way. But in terms of fMRI, this is about as good of a link as we can get in terms of correlating those two things with each other. Okay, so questions, yeah?
So I was wondering about the individual differences and what might be driving those. So do you think I'm wondering if there are strategic differences in the way people encode the information? Are some people better? You're measuring memory explicitly here. There are others you could measure it explicitly. I'm just wondering if you thought more about why some people are sucky at it.
Yeah, that's yeah, I haven't quite phrased it that way, but I'm not sure actually. So intuitively, what I suspect is going on and driving a lot of this is that, and this is totally anecdotal, we've got no data about this whatsoever, is that some people report that they vividly imagine the orientation during the task, and other people say, oh yeah, I said that was like 2:15 on a clock or something. And we try and discourage the latter strategy explicitly by randomly drawing the orientations we're having them remember. So it's actually a very ineffective way to remember is this 46 degrees or 46.5 degrees, right? That doesn't lend itself well to a verbal code. But I do think there are probably a lot of individual differences in your ability to use mental imagery. And that may be one of the driving factors, I'm not sure. That's something we're definitely interested in, and there's actually a population of people, I'm blanking on the name of it, maybe you can help me. But there's a clinical thing that's been defined where people report having no mental imagery whatsoever. And that should be really cool to kind of get in touch with folks like that, but I don't know, we'll see. Yeah?
So the second kind of cue that needs to be rotated, how far is that away from the original stimulus, and how does that have any relationship to how well it's aligned?
Yeah, so they're random with respect to each other. So there's no consistent information from one to the other. I suspect though, and I'm like 90% sure this is true, I'd have to ask one of my students, that when the grading lands close to the target, you're probably a little bit better. But that, I mean, whether that has to do with better memory or reduced motor error, things like that, I'm not sure. But in the experiment itself, they're just randomly generated. So. Okay, cool. Any other questions before I go on here? Okay, great. Okay, so thus far, I spent the first half hour or whatever building up the sensory recruitment hypothesis, right? And presented some data that it's like, activation patterns in V1 are related to behavior and all this kind of stuff. But there's something that's really wrong with those two tasks, and that is we just have this big, 10 or 14 second blank delay period, right? And if you imagine back to the example I gave you at the very beginning of looking for your car keys, that's not at all what's happening, right? So you kind of hold the image in your head of your car keys or whatever, and you start looking all over, throwing the kids' stuff on the ground, and you're getting all this visual input coming into your brain, right? And so recently people have pushed back on that idea in a very logical, well-reasoned argument, and they just said, look, it's really inefficient and probably bad engineering to use the same chunk of tissue to see and to store information in memory, because usually you're making saccades, you're getting that new input, and it's going to just wipe out the memories that you've got stored there, okay? And again, that logically I think that sounds totally reasonable. And there was a study done by Betancourt and Zhu a couple of years back where they actually directly went in and tested this idea, right? And so they designed a study that, actually I'll gloss over a few. It's basically the same thing we've been talking about, where they're asking people to remember an oriented grading They actually do it a little bit differently where they present 2 gradings and then they post-cue you. And so in this case, you get grading 1, grading 2, then they say, okay, remember grading 1 here, okay? But essentially it boils down to pretty much the same thing. You're just remembering a single item, single grading. And then there's an 11-second delay period here. And then they ask you, okay, does that grading match the thing you were remembering or the thing you were supposed to be remembering? Okay? So same kind of delayed match to sample task. But then they introduced a new manipulation here. And they either had a blank delay period, like everything I've been talking about so far, or they presented a bunch of faces or a bunch of gazebos during that delay period. Okay, and I'm not sure what's up with the choice of stimuli here, but I think the point is they're just blasting away at the retina with new input to see if they can overwrite the memory representations in early visual cortex. Okay. Okay, and then the other thing that they did in this study is they also looked outside of early visual cortex and they localized a region in superior intraparietal sulcus. Again, I don't want to make a whole lot about where it is, but I just want to mention it because it's outside of kind of the primary sensory cortices here. Okay? Okay, and so the results of their study are pretty straightforward, right? So in early visual cortex, so they're looking, I guess in this case, at V1 through V4. Just like I showed you before, when there's no distractor during that delay period, they can reconstruct, or not reconstruct, they can decode in this case, what the subject's remembering. So are they remembering, that grading or this grading, right? However, when they present a distractor, so either a face or a gazebo, then that information appears to be wiped out completely in early visual cortex. And so, consistent with kind of what we all probably find very logical, if you're seeing stuff and trying to remember stuff with the same chunk of tissue, it interferes with each other on their account, okay? Now, if you look up in this superior parietal area that they've localized, you find that the difference between classification accuracy when there's no distractor present and when there's a distractor present goes away largely. And really the important thing is that even when there's a distractor present, you still can decode what the subject's remembering in this area of parietal cortex that they've identified. Okay? And so based on that, they put forward the argument that early visual cortex is not in fact important for remembering information. And instead what you're likely doing is, you see stuff, it passes through visual cortex, then it's recoded out of the code transitions, let me say it that way, from a sensory format into a non-sensory format. And it happens that they're finding this evidence for this code up in superior IPS. But anyway, there's this transition of the format of the code into a code that's more resistant to distractor input. Okay? And so even though you're getting new stuff coming in, that code's not overwritten and you can still find that mnemonic information. Okay? All right, so I'll stop again here real quick for questions if there are any. Everybody good? Okay, cool. Okay, so we saw that come out and it seemed totally reasonable. We wanted to kind of get in and play around with it some. So we designed a task, again, same basic thing I've been showing you the entire time. I've just switched the time axis to be annoying here, so now time runs down that way. And on each trial, though, the subjects just remember a grading. There's a very short delay, like half a second here. And then we flicker either Fourier filtered noise, which is meant to have the same spatial frequencies as the grading here, or we figured what better to interfere with the grading than another oriented grading. And so on a third of the trials, we'll have them remember this, and then we'll just put another grading up and we'll flicker it during the delay period for 11 seconds. Let me just make one quick note about this. The orientation of this stimulus and the orientation of the distractor are independent across the course of the experiment, and that's important for a reason I'll tell you in just a second. And then actually on other trials, we just omitted any distractor here, and so it's basically just like the second experiment I told you about. Okay, and then they do a recall task at the end, and that's it. All right, so first thing is to actually verify that the introduction of those distractors is causing a big sensory evoked response in visual cortex, and to no one's surprise, it's causing a huge response. And so if we just look at kind of the average fMRI signal in primary visual cortex, in this case, when there's no distractor, you see there's an initial uptick when you get the memory item, right? And then that rapidly drops off during the delay period, and it's pretty much flat. Okay. When you present the grading distractor during the delay period, you see this nice sustained profile, and then the flickering Fourier filtered noise really drives a big response out of V1 with the bold signal. But anyway, the point of all this is just to say, compared to the no distractor condition here, we're driving V1 super hard in this case, and we're getting a huge response out of it, huge sensory evoked response. Okay. Yep.
What's the time scale on that?
Oh, my bad. The TRs are 800 milliseconds, so this is probably going out to, let's say, 16 seconds. But the delay period right here is about 13 seconds, so that's going to be, from about here to about here probably. And then there's a temporal lag of about 4 to 6 seconds, so you got to kind of shift everything. But this is well into the delay period out here, and actually that brings up a good point too. All of the analyses I'll show you are focused on the endpoints of these time series out here. So. Okay, any other questions at the moment? All right, cool. Okay, so when we do this task, so we know we're driving V1 super hard. When we actually go in and we try and reconstruct now the remembered orientation using the second kind of method I talked about there, so we can get a continuous output. What we see is that there's absolutely no difference between the three conditions. Okay, so if you line them up, they fall right on top of each other. And importantly, going back to what I was talking about the fidelity of these reconstructions, or kind of the bandwidth of them, there's no change in the bandwidth of the reconstructions either. And so what we're seeing is that even when we present this noise distractor that again drives this huge response in V1, the memory signal is not corrupted by that. Okay, and so we're seeing, in our hands at least, that we're able to reconstruct the remembered feature, again, even though we're driving the system with another grading in this case, with another like feature. Okay, and then that's true also in area V4 and pretty much every area of the visual system that we can identify. Okay, so what this suggests, at least in our hands, right, is that there's not, having new sensory input come into the eyes doesn't automatically wipe out mnemonic representations in V1. Okay? And so the next question we wanted to ask, and this goes back to that little side note I was talking about, the two the remembered and the distractor orientation being statistically independent from each other. That provides us the opportunity to actually ask whether we can simultaneously reconstruct both features. Can we reconstruct what you're remembering and reconstruct what you're seeing during that delay period, right? And it turns out that that also works. So using the exact same chunk of data during the exact same temporal epoch of that delay period, we can also reconstruct what the subject is seeing in the form of that grading distractor. Okay? That also works fine in V1, V4, again, pretty much throughout the visual system. Okay? And so what this suggests is that, well, I guess kind of two key points, is that there isn't an obligatory overriding of information, of mnemonic information in V1 when you see new stuff. And second, that you can simultaneously represent mnemonic information and sensory information in the same chunk of tissue. Okay? All right. So actually, let me pause there for 1 second and see if there's any clarification questions, anything? Yep, go for it.
Are you modeling these trials as single trial events, or are you just taking the average pattern from TRs?
We're taking the average pattern from TRs, like, well into the delay period. I have to ask Rosanna, but 7 to 13 seconds or something, let's say. But you can also do this on a time point by time point basis, and yeah, so, okay. Yep.
If you did do this in something like sequences, then you could modulate what is a distractor and what isn't by what you ask them to recall out of the sequence.
Mm-hmm.
So is it possible, do you see something similar? Sorry, this isn't well formed yet. Do you see the memory capacity go up in terms of the amount that they can remember by what you call things, whether you call them things to be remembered versus whether you give it a name, do you cue them toward remembering more?
I don't know about remembering more in a temporal sequence, but you certainly can just present a continuous sequence of gradings, one every couple of seconds in MRI, because we've got to go kind of slow, and just tell them that every new thing you see is now the memory item, or every third thing you see, or anything like that, and then have them kind of search dynamically for something that matches that. And we've done that a little bit also, and that shows the same kind of signatures here. Which is actually kind of nice, because it's also further evidence that seeing an intervening item doesn't automatically wipe stuff out. But But I'll say a few more words about that in a second. So. Okay, so anyway, we've got these two results now, right? So, this other group found that when you present, faces and gazebos or whatever during the delay period, information in early visual cortex drops out, right? And then what we're showing is that if you just present, gratings or filtered noise, two different stimulus classes, things are just fine. So there's no obligatory overriding. And so that kind of left us in this spot where we're wondering, well, what's going on, right? How come we're getting these discrepant results? And even though our logic going into this was that nothing should interfere with the grading more than another grading, it could be actually that gratings and filtered noise are really easy just to ignore. Okay, so we know they're driving a big response because we can look and see, but it could be that people just aren't engaged in that. Whereas if I flashed a bunch of faces or gazebos maybe, people are just super engaged in that, interested in them, and they kind of forget what they're supposed to be remembering, right? And if you think about this, I think at least, or I'll just kind of put this out there, but the idea that you can interrupt a mnemonic representation in the brain should not be surprising to anybody. You can be sitting there thinking, and I could, I won't do it, but you can make a super loud noise, everybody's going to startle and forget what you were thinking about, right? So we know we can interfere with stuff. The question is, what's the key ingredient that makes something a good distractor that wipes stuff out, or that makes it, something you can just ignore and simultaneously represent with mnemonic codes, okay? So what we did then was we figured, okay, look, we're going to use the same design. We've actually done faces and gazebos too, and we get pretty much analogous results here. But I'll just show you one of the two experiments here where we did the exact same thing. So you have gratings or no distractor. And we either had the subjects ignore the distractor, or we had them tell us if there was a slight tilt in the orientation. So a couple of times per trial, the grating would just jump a degree or something, and they had to press a button. Or there would be a little contrast change in a little blob somewhere in the grating here, and they had to tell us when that happened. Okay, so they're either basically attending to the contrast of the grating, they're attending to the orientation of the grating, or they're ignoring the distractor grating. Okay, all right. And what we see in this case, so I'm kind of changing the format of presentation because we're still, actually I apologize for this, you'd never do this in a talk, but we're still analyzing the data. So instead of showing you those reconstruction curves, what I'm doing is distilling the reconstruction curves into a fidelity metric. And we're basically kind of projecting it down onto the angle that should be remembered. So anyway, big numbers here mean that the reconstruction's more narrow like this, and small numbers mean that it's flat, essentially. Okay? So if we look, actually maybe just in V1 to start off with here, when they're ignoring the distractor, we see the same kind of thing that I just showed you, right? So our ability to reconstruct the remembered orientation is pretty fine, okay? But when they actively pay attention to that distractor, let's say they attend the contrast of it, then we get a big drop here in the fidelity of those representations. And when they're attending to the orientation of the intervening distractor, things pretty much, the mnemonic signal pretty much disappears, okay? And that's true across pretty much all areas. So this is going out to V4, but Rosanna's looked out through IPS, and this is also true throughout the visual system, basically, okay? Now, simultaneous with this, so this is now reconstruction fidelity for the remembered item. You can also then go in and reconstruct the sensed item, or that distractor orientation during the delay period. And not surprisingly, what you see there is that if you're ignoring the distractor, our ability to reconstruct it is okay, like I showed you before, but not great. If you're paying attention to the contrast, then you can reconstruct it even better. And if the subject's paying attention to the orientation of the distractor, you can reconstruct it with pretty high precision. Okay? And so basically what's happening here is there's just a trade-off. So when you are paying attention to the distractor, our ability to reconstruct the memory drops out, and our ability to reconstruct the distractor goes up. Okay? Now, one of the key things that happens here also is that behavior gets worse when you're paying attention to the distractor. So your ability to accurately report what that remembered orientation was goes down when information goes down in this condition when information in early visual cortex drops out about that remembered item. Okay? And so, again, this is just showing kind of this push-pull relationship. And I would actually say that this doesn't so much support the kind of first experiment, but it's just kind of a logical necessity, right? Because, again, going back to what I was just saying, I know that I can interfere with your memory somehow, right? And this is kind of a hammer approach way to do it. But what this is showing is that when mnemonic information drops out of early visual cortex, behavior suffers as a result of that. Okay? All right. Okay, so here's just the total speculation point about all these data and how things might work. And this is actually where I'd love to get feedback from people. So in addition to clarifying questions, the floor is totally open now to telling me how stupid this is, or maybe it's on the right track, or anything would be useful at this point. But we've been trying to figure out how these mnemonic signals and sensory signals can coexist and kind of compete with each other in a way that makes sense with the data that I've presented so far. So one of the ideas we've been playing around with is based on some laminar recording data from Peter Rolsema's lab in Amsterdam. And basically what they do is, so they're doing laminar recording. The working memory task is a little bit different, but the spirit of it's largely the same. The animal's trying to remember kind of the trajectory of a line. Let's say it's a spatial working memory task, just for simplicity. But the important thing is that when you present the stimulus to the animal now, or when they present the stimulus to the animal, you get a lot of multi-unit activity in input layers, layer 4 in primary visual cortex, right? But during the working memory delay period, which is shown here, what you see is sustained multi-unit activity in the feedback layers, so superficial and deep layers here, okay? And there's not much happening in these in these input layers, sorry, okay? And so if you take that data and kind of do a massive kind of extrapolation back to what I've been talking about, so if I ask you to hold something in mind, or I ask you, to find your keys, or do any of these tasks that commonly engage working memory, right? Do a math problem. That's a purely top-down feedback signal that's got to be coming in, because there is no sensory signal. Especially if I just say, okay, imagine your keys, imagine what your car looks like, right? No sensory input coming in at all. So no driving input here. Any activation we see has got to, by definition, be due to reentrant feedback into early visual cortex. Okay. And so if that's true, then we might be able to simultaneously reconstruct the contents of memory and the contents of what you're looking at because of this layer division of input and feedback activity here. And so presumably, that sensory distractor, especially when you're ignoring it, right, is going to be driving massive responses in the input layers, but maybe that can simultaneously coexist with these reentrant and feature selective signals into other cortical kind of targets here, okay? Now, the interesting thing about this, though, is that if you ask subjects to ignore the distractor, right, that's just passive kind of sensory input coming into the input layers here. But if I ask you to simultaneously now try to remember something and pay attention to something else, both of those two things require simultaneous top-down input into early visual cortex. And they might be competing more in these feedback layers, and the signals might be mutually interfering with each other and wiping each other out. Okay, so that's kind of one potential idea, and like I said, we have absolutely zero data ourselves about this, but it's just something we've been kind of toying around with. Okay, so just to kind of wrap up and to make sure I kind of, I'll try and explain a little bit about where we stand on this, but so I started off with this sensory recruitment idea, and that memories live in V1 or however you want to say it, and then walked through some very logical pushback on that, which is that's silly, doesn't make sense. You shouldn't be using the same chunk of tissue to do two things at the same time. But I think the data I think are pretty clear on this, and then logically it might also be apparent that things can coexist, right? You could have kind of passive sensory inputs passing through an area, and those could coexist with feature selective top-down modulations to that same region, right? And I also really want to emphasize that I'm not certainly saying that memory is in V1 and that's it, right? I mean, I think that would be ridiculous. If I ask, I give you my phone number, you're not going to remember that in V1. And so the types of codes that we're looking for when we talk about memory, types of signals, whether it's sustained spiking or silent codes and things like that, are going to move around dramatically depending on the format of the information you're trying to remember, what you're trying to do with that information, whether it's a really precise task or a coarse kind of code, and so forth. But I think there's a lot of really cool room here to explore when and why information is transformed from the sensory format into non-sensory formats, how competing top-down demands might mutually interfere with each other. So it might be a good model system to kind of understand why some tasks interfere with each other and some don't. And then also, of course, we want to seek out a better kind of mechanistic understanding about how the coexistence of memory and sensory signals might actually play out in early visual cortex. So, and again, any ideas are totally welcome on this front, and I would love to hear what you have to say. And then before I wrap up, I just want to acknowledge the people who worked on all these projects, especially Rosanna here. She's a postdoc at UCSD. She did all the second half of the talk basically and a whole lot more. She's been really fantastic. And yeah, that's all. So thanks. Yeah.
Has anyone tried a TMS application to these regions and see that effect?
Yeah, so people have tried that. And I would say the results are a mixed bag. It's also harder than it sounds. So I mean, it's fairly easy to get the foveal confluence with TMS, I think. And actually, maybe you can speak to this even better than I can. But to get stimulation in the peripheral representations at all is much more challenging. And again, the results, I have a sneaking suspicion there's a big file drawer on this. And there might be a lot of null results out there. But the results that have been published, I would say, are largely in favor of the idea that that interferes with memory, but not entirely. So, I don't know, open question. But yeah, great idea. I wish it was just like you could just, zap somebody and make it go away, but it doesn't seem to work. So, yeah.
I think you said one of the last experiments that you only showed the data for V4, but IPS showed the same pattern of this trade-off, right?
Yeah.
But I thought IPS was the recoding area.
Yeah. So I was glossing. Yeah. So I'm glad you brought that up. So the memory. Yeah. Okay. Right. So this is going to go a little bit into the weeds. So sorry, but so all the models that I trained, or that actually Rosanna trained, I didn't train anything, were done that I showed here. Were done using a sensory task, basically. So they're just looking at a grading. It's just blasting away for 5-10 seconds at a time, right? So if you train the model that way, what you see is that transfers, a model trained that way will generalize very nicely to remember to mnemonic patterns in early visual areas, not so great to IPS. Okay? If you take the memory task and train the reconstruction algorithm on that, then that generalizes beautifully in IPS. And so what that means is that there is mnemonic information. It's basically like Yauda was arguing there. And yeah, I'm totally on board with that idea. So if you train it on the memory task, IPS works beautifully, which means that it's got mnemonic information, but it's not in a sensory format. If that made sense. I don't know. Sorry, it's kind of a hard thing to explain, but yep.
Sorry, you may have talked about this in the last bit. I could have missed it, but if imagine going from V1 to early visual areas and looking at sensory representations and then recoding. Mm-hmm. And then if you're asking an individual to imagine visualizing something, does it activate V1 the way that is true to the true sensory input? Mm-hmm. And if so, that then implies that somehow the recoded version has to get sort of re-expressed as a visual.
Yeah. Okay.
Early sensory representation.
Yeah. So this is a great question. So what I would argue is that probably what's happening. So if you know in advance that you're going to have to make a really fine discrimination on something, then I suspect V1 stays online or involved in that the entire time. And if we can knock out V1 activity by, in effect, banging, making a loud noise, doing something that interrupts your memory, and if we can get V1 to drop offline, your performance suffers. Okay, so you can still do the task, but you can't do it as well. And so what I suspect's happening here, and this is actually exactly the direction we're going, is that if you take the information in, it is being recoded. We find evidence for that too. I didn't really show it here. But if I take the high precision representation offline, you're going to suffer. And if you just have to rely on that recoded information, performance is going to be worse. And so I don't think necessarily that there's, it's a strictly serial process. Like, you get high precision sensory coding, you recode it, and then if you are asked to do a high precision task, you unrecode it, right? I don't think that's what's going on because behavior suffers. And people don't seem to be able to undo that. I think what makes a lot more sense is that there's information compression happening, and you're getting a coarser and coarser representation as you compress that code more and more, do more and more recoding. And if I force you to rely on that compressed code, then you're not as good at the task. So, yep.
I think this follows from what Jared was just asking, but with this, to kind of test what you were just saying, could you just test memory difficulty? So is it just a match, non-match system?
The very first one I showed was match, non-match. The rest are recall. In our hands, Yauda's experiment was a match, not match.
So
Right.
you really have to kind of have a high fidelity representation to be able to do
That's the idea, right? And so, this is actually a really interesting thing too, because most monkey neurophysiology is like remember one of two directions of motion, remember maybe four directions of motion. But if I ask you to do that, you wouldn't use a visual code at all. You just say, oh, it's up, it's down, it's left, it's right. And so that kind of memory might be qualitatively very, very different than the kind of thing we're doing here, because we're really trying to force people to remember the visual details and not rely on a verbal recoding scheme or some kind of digital code in this case. But. All right, cool. Well, thanks very much again.